Those of you who've been on the earlier meeting and those of you who have just joined us, welcome. It's to the Neuropsychiatric Disease Working Group evening talk. It's with great pleasure that I introduce to you Tychele N, who's an assistant professor at the Department of Genetics at Washington's University School of Medicine in St. Louis. And she'll tell us about the Novo mutation in an enhancer of EBF3 in simplex autism. Tychele, thanks, and take it from there.
Thank you so much, and thank you for that kind introduction. I'm just going to put my slides up here. And is everyone able to see those okay?
Yes.
Okay, perfect. So today I will be, as mentioned, focusing on de novo mutation in an enhancer of EBF3 in simplex autism. My talk is actually a little broader than that, though. I just wanted to first give you a little bit of the background of autism and the genetics of autism. Then I wanted to tell you a bit about the whole genome sequencing efforts going on, and then dive into the de novo mutation work in this enhancer. And I thought we could end on some thoughts in future directions, since it said the end of the session would be questions and discussion time. So autism, I'm sure this audience doesn't need a deep background, but it is a neurodevelopmental disorder with an onset in childhood. It has a high heritability. It's fairly common in the population with a prevalence now of 1 in 54. And it's biased towards males. So about 80% of all cases are male. And I think this is really an intrinsic feature that we should think about in terms of autism more generally. So we and many others, and many of you actually on this call, I'm sure, have been involved in understanding the genetics of autism. Our first real wins were in the discovery of single gene syndromic forms like Fragile X syndrome due to mutations in FMR1, Rett syndrome due to mutations in MECP2. We now know that there's a really strong contribution from common variation, both from genome-wide association studies and polygenic risk scores, and that their contribution could be up to 50%. And yet we stand at a point with just 5 genome-wide significant loci. I mean, it's been a lot of work to get to this point, and it's, I think, very exciting future, but that's kind of where we are at this stage. I would argue that in terms of gene discovery, the study of rare variation has really fueled the field forward, both in the discovery of copy number variants and de novo gene disrupting mutations. So copy number variants, including things like 15q11 to 13 maternal duplications, 16p11.2 deletions and duplications, and many others. We now stand at a time point when we know greater than 300 genes implicated in neurodevelopmental disorders, including autism, due to mutations that are protein coding and de novo and gene disrupting. So these are things like CHD8 and DYRK1A and others. However, there is this considerable overlap of these genes involved in intellectual disability, and so it's been sort of hard to disentangle what are autism-specific types of genes. Nevertheless, we can proceed forward and begin to learn about the clinical side of things. For example, once we know a gene, we can go back and look at patients, like Bernier did in 2014, and this was probably one of the earliest studies doing these genotype-phenotype correlations based on protein coding de novo discoveries from whole exome sequencing in autism. And what they found when they went back and looked at these individuals is that they share a constellation of features in addition to autism, so macrocephaly, sleep disturbance, and gastrointestinal dysfunction. And just in the last year, we published a paper with clinical collaborators diving into this further, where we took the de novo gene disrupting variants and split them into genes that are either significant for, statistically significant in autism, actually be more appropriate here to say in neurodevelopmental disorder genes or not, and then whether they were in CHD8 itself, which is a chromatin binding gene, or in a target of the CHD8 protein, or not in a target. And it turns out that when we begin to look at the phenotypes of people who have mutations in the same pathway, as we might expect, they do share a number of features that are not shared when you look at individuals who have mutations outside of that network. I just wanted to put in two quick slides on the sex bias and autism. It's an area I've worked on quite a bit, and I think that it's appropriate in the terms of the background and neuropsychiatric disorders more generally. I think that probably most people in the autism field are really well aware of this bias and that it's really important. But I think as a neuropsychiatric disorder working group, we can think much more broadly than that. And when we look in the DSM-5, for example, and look at the fraction of cases that are male, as shown here on the Y, and then the different disorders on the X, we can see some things are biased towards females, like anorexia nervosa. Some things are biased towards males, like intellectual disability and autism spectrum disorders. Now, it could be ascertainment bias, it could be diagnosis, or it could be something biological or a combination of all of those items. So we and others have identified that when we look at protein coding de novo variants, there's a greater enrichment in females than in males. And this has just been an observation that's consistently showed up in papers. And in the last year, we went back and actually reanalyzed all that data and looked at it by males and females and asked, is the discovery of protein coding de novo variants, so rare variants of high effect, are they really, greater discovery in females? And so here I'm showing you the Venn diagram of what survived after replication, and just noting that this male sample size here was twice that of the female sample size. And you can see that the gene discovery in raw numbers is about the same, but because our sample size was so much smaller with females, our gene discovery was greater. But it was hard to disentangle that again from the question of the intellectual disability, since the rate of intellectual disability was higher in females. There were just a few genes which were really specific to females, and those were on the X chromosome, and they were in general escapers of X inactivation, meaning that they're expressed at about twofold expression level in females versus males. And when we looked at the genes that were enriched, they were enriched for processes like chromatin binding transcriptional processes and being localized to the nucleus, which is a pathway that we're very familiar with in autism. But I just want to take one moment here and just stop and think, when we get to the non-coding section, I want you all to be thinking about what would we expect of a non-coding mutation in autism to look like if we have this enrichment for protein coding in females. And I think it's something we need to explore as we move forward as a field. So I'm going to now dive right into whole genome sequencing in autism. And I wanted to just take one moment to really acknowledge the people who've done all the work of generating all this whole genome sequencing data. So I think about 5 or 6 years ago, the NHGRI and the Centers for Common Disease Genomics and the Simons Foundation came together to basically fund the sequencing of many, many individuals with autism and their families at the New York Genome Center. And I just can't emphasize how groundbreaking of a study this really is. Previous work in autism has sequenced many families, but at different, for exome sequencing, for example, at many different centers. And that's great, except for when you start to look and each center has sort of different requirements of what they think is good QC and what is important to do for analysis. And what happened with this approach was that everything was done at the New York Genome Center, from library preparation to the initial analysis. And that has been a huge benefit to the field in terms of studying autism genetics. So I think that this is the largest whole genome sequencing collection of individuals with autism in the whole world. I think the number is now in the tens of thousands of genomes. The sequencing primarily focused on DNA extracted from blood, so we don't have to worry about cell line artifacts when we're looking for de novo mutations. There were standardized sequencing workflows, uniform computational processing, ability to do those sample overlap checks before distributing to the community. The data, I will not show you the raw data today, but it is of exceedingly high quality. It's just really beautiful data. And many of the data sets are already available through things like Anvil and Safari Base. So you too, just like me, can get your hands on that data and start asking the questions you might want to ask. And there are already, by my count, just looking this weekend, greater than 30 papers and preprints utilizing this data. So I think the path is, and the future is very bright in terms of our study of genomes in autism. So we have been primarily focused on a cohort called the Simon Simplex collection. I think this is the largest sequenced simplex autism cohort for genome sequencing. This is actually the 10-year anniversary of the collection, which just really fascinates me that it's been around this long. And it was initially hypothesized to have de novo variants, and that's because of the family structure, which you can see here on the left, where we have parents who do not have autism, one child who does, and one child who does not. So over those 10 years, array and whole exome sequencing has been applied, and then now whole genome sequencing. But from those early efforts, we could explain 10 to 30% of all of the individuals with autism based on large copy number variants and those de novo protein coding variants I mentioned at the beginning. So then a few years ago, as I mentioned, whole genome sequencing was done to look for things that might have been missed because of technological differences in the exome. So genome can do a little better at certain classes of variation than exome, even within the exomic regions, but also to really dive into that non-coding space, which we had really not explored as a field. So I'm going to show you a little bit of data from our paper in 2017, and this was our comprehensive genome workflow at that time, utilizing a lot of cloud infrastructure, where we did everything from QC metrics, mitochondrial genome, structural variants, SNVs, and indels. I would argue that our comprehensive genome workflow looks much more comprehensive now, but this is what it looked like at the time. And we really focused on 516 families and de novo variants, so about 130,000 de novo variants. These families were pre-selected to not have loss of function mutations or large CNVs. So we thought this is a really good area to look for non-coding variation. The first thing that we noticed is that when we look just restricting here to only the autosomal variants, there is no overall difference in the number of SNPs or indels in between probands and siblings. That's exactly what we would expect. We don't think that there's some huge difference in burden genome-wide. We think it's actually in specific functional categories, as has been shown in the exome work. Focusing in on the de novo enrichments, one class that we looked at were severe missense mutations, defined as having a CAD greater than 30, and small exonic deletions, which were both enriched. This was 2017, and we were not exactly sure where to look in non-coding space, so we decided to keep it really restricted, and that was to look at untranslated regions, which showed this nominal enrichment, and transcription factor binding sites residing within fetal central nervous system DNase I hypersensitive regions. So basically open areas of the genome that could be potential regulatory. We didn't see significance until we broke those down into fetal promoters and embryonic enhancers. So as this was still quite the early days for non-coding in autism, we were wondering what might be the functional kind of test we could do, because we weren't sure what to anticipate of an enhancer variant. So we teamed up with Dr. Len Pinocchio and Diane Dickel at the Lawrence Berkeley National Lab. And we tested a few enhancers, of which one I'm showing you here today is this HS737 enhancer, which I will come back to a number of times in the rest of the talk. So this is an assay, and it's what they use for their Vista enhancer browser. And how it works is you basically have a reporter sequence, so this enhancer sequence here, and you ask, can it drive expression of a target gene, which in this case is LAC-Z, at E11.5. And wherever it does that, it will light up blue. So for this particular enhancer, it normally drives expression in the midbrain and hindbrain and not in the forebrain. But when we introduce this single G to A change within this enhancer, we have a reproducible expression showing up in the forebrain while still maintaining the old pattern of expression, which indicates that in the first kind of enhancer we really looked at, we saw a gain of function, and I think was quite surprising to us. So a lot more on that in the next section. We're just gonna highlight a couple other points. One was that we had the first opportunity to really begin to look at de novo variants no longer by class, right? Because that's okay, but we could actually come up with something, I would say, akin to a polygenic risk score, but now we're looking at like rare variant risk scores. And how we did that was we essentially took all of the categories that were enriched on their own and then asked, what is the enrichment in probands and siblings of having no mutations 1 mutation, any 2 mutation, any 3, and so on. And because we had quads where we had matched probands and unaffected siblings, we could do this quite nicely. And what we saw was that at 0 and 1, the siblings predominated. At 2, the probands and siblings were about equal in number. And then when there were 3 or more variants of interest, we really saw this enrichment in probands. We wondered what this could potentially be. And so we focused in on these genes affected here and found that with cell type specific expression analysis that these were enriched for D1 and D2 positive spiny neurons in the striatum. And that's a category that has a cell type that's been implicated now in a number of other studies as well. And importantly, we never saw any enrichment in the unaffected siblings. We also looked at autism genes themselves, and you can imagine back in 2017, I was looking at a gene list of 57 genes that were statistically defined for protein coding de novo. It's amazing to me that we're now at greater than 300. But at that time, we saw enrichment in the 57. We also saw enrichment in Safari base, which is not just statistically defined, the Safari gene list. When we looked at Safari gene in more detail and asked, do the probands have multiple hits or do the siblings have multiple hits in those genes, we saw enrichment there as well, in overall, in non-coding as in coding. I think to me, even though I'm really interested in non-coding, when I first saw this data, the most interesting thing to me of this data set was the protein coding result. So this indicated that we had 10 probands where they had mutations in at least 2 different autism genes, which could potentially be having an effect. And we didn't see any siblings like that. Well, we could argue quite a bit about how we might estimate the contribution of each of these classes of variation. What we did at the time was to use something called an attributable fraction estimate. And this is a way to look at a certain category and say the percentage of cases that can be explained by that category is X%. That's like the attributable fraction. But because we were sitting at a time then where we only had 516 families with genome data and we didn't have the whole cohort, we did the attributable fraction estimates for these known categories using the 1786 quads that only had exome and array. And we showed that, as expected, de novo likely gene disrupting, you may call them loss of function as well, mutations had enrichment, and they were explaining about 6% of cases, de novo CNVs about 4%, inherited CNVs not stratified by parental origin had no effect, and then multi-hits were about 1%. When we added in our genome data, we saw that for the new categories, so small exonic de novo deletions, severe missense, and multi-hit non-coding, they each contributed about 2%, but you can see the confidence intervals are quite big here. So this suggested to us there is a path forward, but we need more genomes. And I want to point out to you that while I'm so delighted that we had the opportunity to work on this and we're working on it still, many other individuals, including I'm sure many on this call, are also working on this. And I want to show you a few papers from the last few years, and I've just picked one from each year that I think is quite interesting. And I just want to note that each of these papers is utilizing this data from the New York Genome Center. And I will just be keeping our data point on the left hand of the slide for reference. So in 2018, Stefan Sanders' group actually did this really cool study where they applied some machine learning to look at promoter categories, and they found that there was an enrichment in cases for mutations in promoter regions. In 2019, Olga Troyanskaya's group did this interesting machine learning approach where they tried to score variants both in DNA regulating regions and RNA regulating regions. I'm only showing the DNA here, but the RNA also had enrichment. But they were able to show that there was greater enrichment in the probands. And then probably my favorite paper to come out in the last few years is this paper by Markinskoff Papadimitriou. And I think that perhaps Stefan Sanders, he's the geneticist on this paper, so I'm kind of guessing he may have done this analysis. But this analysis really, it floored me. I just thought this is so clever, and I'm really enjoying reading this paper so much. In this paper, what they did was something very, very clever. So they looked in regions of open chromatin, and they looked around autism genes. And you can see that here with the X's. And they see nothing, right? It's not very interesting at all. And so what they did was they applied a machine learning approach to take advantage of that VISTA database that Len Pinocchio's group has very carefully curated over the years. And what they did was they said an element is a positive element if it's a VISTA enhancer that can drive expression in the brain. And it's not a positive element if it's a VISTA enhancer that can drive expression anywhere else, so outside of the brain. And they called these regulatory elements, these PREs. And when they applied that, they saw that there was enrichment around autism genes for all non-coding as well as those within intronic space. And so I just quote this from their paper where they say, although PRE containing categories did not cross the threshold for statistical significance, they had higher relative risks than the same categories using OCRs rather than PREs. This demonstrates the value of our machine learning approach for identifying a subset of OCRs related to ASD. So what they're actually doing, right, is they're not just looking at open chromatin, but they're asking, does that open chromatin region also contain the sequence context required to drive expression in the brain? Why I thought it was so cool is because for the last year in my independent lab here at WashU that I started, I was thinking about VISTA. I've been focused on VISTA so much because we have the evidence in vivo that they are actually enhancers. And I think that in non-coding world, the burden of proof is so high. First, normally you would first have to find some genetic signal. And then once you find that genetic signal, you have to ask, okay, is that enhancer really an enhancer in vivo, or is this some kind of artifact? And or what time point is it an enhancer? Like maybe it's a time point we can't profile at the moment. So I've been focusing on the VISTA approach, and I thought, wow, this paper is so cool. I think we're also on the right track. And I just point back to this HS737 VISTA enhancer from our paper that I mentioned earlier. So before I dive into the EBF3 enhancer story, I have one final background slide that kind of leads into this, and that's that the last year I worked in my postdoc, which is now 2 years ago, I focused on creating a de novo workflow that would give us a high specificity so that we wouldn't have to worry that the variants maybe are not real. In some of my previous work, we focused on sensitivity to really get at all the regions of the genome, but here I was very focused on specificity. So I created this workflow leveraging all of these different callers on that 2,671 family data set, and then did a series of filtering steps and a conditional inference analysis on over 3,000 sites that were validated by Sanger or PacBio sequencing. And came down to a final de novo call set with a 99.5% validation rate. That data set has been integrated in this preprint by Amy Wilfert in the Eichler lab. And so that is the data that I will be using in the remainder of the talk. So I want to first point out that this work is being led by Evan Patty, who is a graduate student here at WashU in my lab. And he's just doing some amazing work. And we've been able to put this up on bioRxiv already. And I really, since this is not published yet, I would really welcome any feedback on this work, either today or by email. So based on the HS737 observation and based on the Markinskoff Papadimitriou paper, we were very, very focused on these VISTA enhancers. And so we worked with some collaborators to adapt the FIT-DNM model, which many of you may have heard of, which normally works on protein coding space and looking at de novo mutations, but we adapted it to work in non-coding space. And then we applied it on all of the VISTA enhancers that are able to drive expression in the brain in vivo, so the VISTA data set. This is the table of the results. I'm only showing you the enhancers which have at least 2 de novo mutations in the full 2,671 families. And I also have split them out by discovery set, replication set, and the combined data set. And this is a moment I just want to step back and tell you, in 2017, when we tested HS737, with Len, we didn't have any of the new genome data. I mean, there's no way that we could have predicted that this enhancer would be the top enhancer today. In fact, when it came out, I was kind of like, this is unbelievable. I just can't believe it. But it is what it is. It is the only enhancer that has significance in discovery, replication, and in the combined data set. We have a number of other interesting candidates here, but 737 is the only one significant in all three. I did want to point out just quickly this HS2333 element. I'm not going to come back to it again today, but it is an enhancer in the intron of DYRK1A, and it is known to bind to the promoter of DYRK1A in the human hippocampus. And it also has 3 de novo mutations, just like HS737. And it's a very interesting enhancer. To me, it's interesting because DYRK1A is a genome-wide significant gene for protein coding de novo mutations. But let's focus in on HS737. So I'm a human genomicist / geneticist first. So the first thing that I wanted to do before we moved on to anything else was to look for further genetic evidence that this might be important. So we turned to copy number variation data in the form of this database called Morbidity Map, where we have 30,000 individuals with neurodevelopmental disorders, 20,000 control individuals who've all been assessed by microarray. And what you can see here is that there were 27 individuals with NDDs who had a deletion of this enhancer versus no controls, and 6 who had a duplication versus no controls. So we didn't have a lot of additional genome data at that moment beyond the 2671. And so I thought, why don't we try to genotype more controls? So we came up with a read depth genotyping method using the QuickMare2 tool and a little bit of downstream analysis that we've worked on. And we benchmarked this read depth genotyper against Sanders' paper in 2015 and found that we can find 97.3% of all the de novo structural variants that we should find. Another 2% we found, but they actually look mosaic based on the genome data. And then 0.9% didn't validate. And we haven't explored that too much further. But we were thinking this is probably a pretty good read depth genotyper, but as you're aware, those structural variants are quite big. So we went back and asked, what if we only genotype the exons of genes where we know there are these big copy number variants, right? And so we focused in on 16p11.2. We looked at this gene KCTD13. Here I'm showing you the copy number on the Y and then the position on the X on chromosome 16. And then these are each of the exons. So we only assessed the exons, and then we looked at the read depth, and we just plotted that with lines here. So the dotted line is the deletion cutoff, and here on the bottom, and the dotted line here is the duplication cutoff. And we found that when we just looked at the exons, we were still able to recover these deletions and duplications that we were expected to find. So we then applied this to additional controls. We looked at the 1,000 genomes data, high coverage genome data that also was recently sequenced by the New York Genome Center. And we found there were no individuals with deletion of this enhancer, no individuals with duplication. Looking at the unaffected mothers and fathers in our study, again, no deletion or duplication. So when we add this with Morbidity Map, this suggests that there are 56,256 alleles that we've looked at, and this enhancer is never deleted or duplicated. And this indicated to us that it's perhaps a dosage sensitive element in the genome. And for good measure, we checked NOMAD to see if there had been anything found there, and there are no CNVs reported in this region in NOMAD either. So then we took a look at the families that had a regulatory mutation in one of these, in this enhancer. And the first thing that I want to point out, which was sort of surprising to me, was that all 3 cases were male, and none of them had intellectual disability, and all 3 either had motor problems and or hypotonia. When we looked at the other variants found, so these are all de novo mutations that were either missense or deletion, again, missense here, we scored those with ACMG criteria, and none of those were considered to be pathogenic. So it seems like they're basically individuals who are male, not intellectually disabled, and don't really have any known genetic cause. We also found one NDD patient in the Decipher database where we could go back and look at the phenotype data of a non-coding deletion, and that person, the two phenotypes listed were autism and motor delay. So we'll come back to this hypotonia thing a little bit later. I was actually really curious about the polygenic risk scores of these individuals. And so we reached out to Elise Robinson, who put the polygenic risk score data from the Weiner et al. Paper in Safari Base so that we could get access to that. And I just thank her immensely for that. We were really interested in the ASD polygenic risk score. And this is the proband distribution. And each of the dotted lines is an individual, one of our 3 people. And as you can see, they're all sort of in the middle of the polygenic risk score for ASD. And so it's probably not a super big contributor in these individuals. They're on the low end of the schizophrenia polygenic risk score. And then their educational attainment polygenic risk score is stratified somewhat by their IQ. So it seems like maybe the ASD polygenic risk, though, here is not enormous in these particular individuals. So we then turn to the HS737 element itself. HS737, based on psych and code data, is a H3K27 acetylated data enhancer. It is central nervous system DNase hypersensitive, and it is highly conserved. When we turn to the mouse encode data to look at the enhancer in more detail, we looked at the chromatin state track across many different tissues, as shown here, and many different time points from E11.5 to P0. And as you can see, this yellow coloring is all lighting up in the brain regions and not in the other tissues. And you can see in most of them it's active. So it seems to be a brain-specific element. As I flash back to our study before where we saw this gain of function, we noted that at E11.5, the enhancer itself is in the poised state. It's not fully active, but it's poised, which could be why it has this gain of function that's kind of leaky. Looking at attack-seek data across E11.5 to P0 and H3K27 acetylation from E11.5 to P0, we can see this element is very active during embryonic time points, but then by the time we get to birth, it's basically shut down. So this is very preliminary data from Len's group. He's actually taken this enhancer and mutagenized it with 2% mutagenesis or 5% mutagenesis just to see what happens if you start to mess with the enhancer. So again, the wild type pattern is this nice crown-like pattern. This particular 2% mutagenesis allele has a gain of function here, and it's a little bit stronger here. These 5% have complete loss of function. This one has a partial loss of function. This one has a partial loss and a partial gain. So it seems that just with the dosage sensitivity information might suggest that we probably do not want to mutate this element. So Evan in my lab did some luciferase assays in Neuro 2A, and one thing that we made sure to do was to always have a positive control. So we use this RET +3 enhancer, which is well known in Hirschsprung disease as a strong enhancer, just to show you that the assay is working. When we focus in on the relevant data for this particular experiment, we can see that the wild type enhancer seems to be a weak enhancer, and all of the mutations are differentially driving expression from the wild type. So they all seem to have some effect. This really puzzled us. It's cool, but very interesting. And we wondered, is there some potential common mechanism in the sequence? So we focused in on each of the mutations, one here and another one and another one. And we're showing you the reference sequence on the top, the alternate sequence with the mutation shown in red, and then the conservation. And as you can see, each of these positions is conserved all the way to frog. And what you might appreciate immediately that we didn't appreciate until after looking at it for a bit was that each of these mutations seems to disrupt an AAT motif. These are not the same AAT motifs. These mutations are in different places in the enhancer. But what you'll notice here is we have an AAT going to AGT, which is taking a SOX family member from the bound to unbound state. So we've disrupted that AAT. Here we disrupt the AAT again, so AAT to AAC. And that's predicted to take an ARX family member from bound to unbound. And here, This is the gain of function mutation. It's a AATG to a AATA. So it's actually allowing the AATA, the stronger site for an ARID family member, to come in and actually bind there. But I think it's quite interesting to look at that sequence context. So with all that information in mind, we just take a step back and say, yes, it's an interesting enhancer, shared phenotypes and everything. But what in the world is this enhancer doing? So the first thing we looked at was the topologically associating domain that it resides in the genome, because we think, right, that enhancers should regulate the gene. The gene it regulates should be in the same tag. And I think half of you will go, ooh, there's no genes nearby. That's so lame. And then the other half of you will go, wow, there's no genes nearby. That's really cool. And I guess we're more like that. We thought that's really neat. There's nothing there. It's going to be really hard. So the first thing we turned to was actually human mutation data. And we looked at NOMAD for what genes might be constrained in the region. And the only gene constrained is this EBF3 gene. And mouse RNA-seq data from ENCODE at the same time point has the same expression pattern as what the enhancer 737 does, where it drives expression. So we thought, this is really interesting, but what can we do? And this was actually not easy. It took us a long time, and we joined up with some collaborators trying to figure out what can be the gene that this enhancer is targeting. And so we turned to this Bonev paper and reanalyzed it, looking in embryonic stem cells, neural precursors, and cortical neurons. And this is a Hi-C data set. And what you can see, the enhancer is here, and then the EBF3 promoter is here. You can see in embryonic stem cells, there's no interaction. In neural precursors, we begin to see an interaction. And then in cortical neurons, which I've made bigger here so you can really see the enhancer and the EBF3 promoter, there is an actual computationally called loop in the Hi-C data showing that HS737 is indeed targeting the EBF3 gene. So at this point, we have EBF3, and I think the name is not that interesting. It's not telling me a lot. I was like, man, why wasn't it CHD8 or something really obvious? But I began to look more at EBF3, and as it turns out, EBF3 is actually a very interesting gene. So it is genome-wide significant in both this Ko et al. Paper from the Eichler group and the Kaplanis et al. Paper, which I believe actually is now published fully from GeneDx and DDD. And that's a beautiful paper. And what they found was that there was enrichment of missense and loss of function mutations, which I'm showing you here on the linear diagram of the protein, and also on when we modeled it on the 3D structure. We went, for good measure, we went back and reanalyzed those 37,692 trios, the de novo data from that, and with the chimpanzee human de novolizer, these commonly used tools for assessing de novo enrichment, and saw that, of course, they're enriched for missense and loss of function mutations. And EBF3 itself is a transcription factor, and many of the mutations reside in the DNA binding domain. As you read more about the gene, though, it's actually involved in this thing called hypotonia, ataxia, and delayed development syndrome, also called HADS. And that was so intriguing to us, given that our patients also have this hypotonia phenotype. We were able to identify some ChIP-seq data from SKNSH cells for EBF3, and that's a neuronal cell line. We took that data and remapped the peaks to promoters in the genome and found that about 16% of all genes in the genome are bound by EBF3, and that it actually has a preferential binding. There's an enrichment for binding at other neurodevelopmental disorder genes from these two papers that I just mentioned. But when you begin to look at that list, EBF3 is binding the promoters of genes involved in chromatin binding and DNA binding. So that one category, it's binding genes like CHD8, CHD2, ARID1B, but it is not binding the synaptic category of genes like SCN2A, which kind of surprised me that it was much more specific for that branch of the autism pathway networks. EBF3 itself is expressed just about everywhere. This is from the GTEx project and the Protein Atlas. And these are the brain regions here. This may be why protein coding mutations result in the HAD syndrome, whereas our brain-specific enhancer seems to be having a much more subtle phenotypic consequence. So I put together a proposed model for understanding genomics of neurodevelopmental disorders. It's somewhat simplistic and it's based only on our work in this presentation. And of course I put diamonds because I'm not sure if things are turning things on or off at the moment. But if we just focus on this network, we can see that EBF3 basically sits here in the middle. And we have these upstream regulators like SOX, ARX, and ARID5. We have downstream genes like CHD8, ARID1B, CHD2, and many others. These genes themselves actually regulate many other genes. And then we have our little enhancer coming in here and doing brain-specific regulation. When we begin to look at the phenotypes of individuals with protein coding mutations, in particular in these higher network genes, it seems, this is a qualitative assessment here, not quantitative, it seems that the higher network individuals have more severe phenotypes like lissencephaly and ARX, HADS and EBF3, CHD8 would be this autism, GI dysfunction, sleep disturbance, ARID1B, autism, intellectual disability, CHD2, autism, epilepsy, and then HS737, autism and hypotonia. I'm just so excited to see where the future is going in this work in terms of the field more broadly, but I think once we place all of these genes back into their respective networks, we're going to learn so much about where biology of autism, but also where non-coding sits within that biology. So in conclusion, whole genome sequencing in now almost 2,700 families has identified enrichment of mutations in this HS737 enhancer. We see that we also have enrichment of copy number variants in individuals who have neurodevelopmental disorders. We see dosage sensitivity in controls. We see shared phenotypes in individuals with de novo mutation in the enhancer. The enhancer itself is brain specific with highest activity before birth. Random mutagenesis really affects the enhancer itself. In luciferase assays, the patient mutations do have a differential effect from wild type. They all disrupt an AAT motif, and that is predicted to either disrupt or add transcription factor binding sites. In the TAD we looked at, the only constrained gene is EBF3, and indeed Hi-C data confirms that that is the interaction. And I should note that this interaction in the human genome is occurring at a distance of greater than 1.4 megabase pairs. So this is really a long-range interaction. EBF3 itself is genome-wide significant for protein coding de novo variants and is also involved in this syndrome called HADS. Its transcription factor binds many other NDD genes. It's widely expressed, and that may be why the phenotypes of protein coding are more severe than non-coding. So I want to just end with some thoughts in future directions in terms of the future of non-coding work and the genomics of autism a little more broadly. And the question that I pose to my student and that I pose to others in other calls is, what if 10 years ago, the decision had been made to do whole genome sequencing on the Simon Simplex collection? And I think if I had been in that room, I would have been jumping up and down saying, yes, let's do it. We've got to do it. We've got to do genomes. And I think that the people who are sitting in that room, or probably many rooms, made the right decision because when we look at our work today, we find that the Vista Enhancer browser, yes, it did exist at that time point, so we could have looked at Vista elements. However, there was no gene list of genome-wide significant genes in neurodevelopmental disorders, and that has really been fueled by whole exome sequencing. So we would have never known EBF3 was an important gene. NOMAD was not up and actionable like it is today, so we wouldn't have had constraint metrics. Morbidity map itself was, I think, probably close to publication, but not anywhere where it is today, where we can just go in quickly and ask, do we see enrichment in neurodevelopmental disorders or not? And the mouse encode data in particular, I know we use some of the psych encode data. We haven't dug in as far into that, but at least for the mouse encode data, it was nowhere where it is now, where we have all this chromatin state information, E11.5, 12.5, 13.5, all the way up to P0. And it's just a beautiful data set to profile these kind of enhancers. So we would have probably been pretty far from finding things if we had done that 10 years ago. So I'm really happy that we're doing it now, because now is the perfect time to have this data. We know that we need many more genome sequence individuals. We do. And we also need a gold standard for multi-test correction p-values. And I have talked to some people and they push back on this, that's not going to be possible. And I agree it's going to be difficult, but I think it's important because for things like GWAS and exome sequencing, we pretty much know where that standard is, right? We know we have to hit a certain p-value. And I think that if we don't have this for whole genome sequencing and non-coding, it's going to be very problematic. So one way we could think about this is building gene-specific models. That in turn depends on knowing the full suite of regulatory elements for each gene. And so I'm not sure at which point we'll be satisfied that we know the full suite of regulatory elements for any given gene, but I think we've come a substantial way so far. The next question is, what phenotype do we expect of an individual with mutation in enhancers? I was surprised that all the patients were male and didn't have intellectual disability, and thinking back, maybe I shouldn't have been surprised. But I also do know we have a very small sample size here. So this is a question that we must, empirically examine in the future. And then what will be the distribution of loss of function and gain of function variants? And I think some people have taken some nice looks at that. So there was this enhanced yeast-1 hybrid assay looking at all the non-coding disease mutations in the human gene mutation database, in this paper in Cell in 2016. I was just fascinated by this paper that most of the mutations resulted in a loss, but there was a substantial number that actually caused a gain. Looking at Evgeny Kvan's work, he's just an amazing scientist as well. He did these 2% mutagenesis alleles of this ZRS enhancer, which many of you probably know, involved in polydactyly and limb issues. And what he showed was that when you begin to mutagenize this enhancer, 44% of the time you're reducing the expression, 26% you have a full loss, 7% of the time you have a gain of function, and the rest of the time it's normal. So I think just saying we have a non-coding single nucleotide variant, it doesn't really tell us much. We have to figure out what it's doing. Is it bringing a new transcription factor in, or is it taking one out? And so the last point that I want to make is on the genome side, I think that we're very, very close to having fully resolved precision genomics for precision medicine is what I would call it. Many people use these terms, but we're at the stage where we can begin to resolve complete genomes. So this is our first neurodevelopmental disorder patient that we've looked at with PacBio HiFi sequencing. So this is now the long read sequencing. And you can see these are the contigs in this bandage plot. We have NG50, so greater than 50% of the genome is represented in contigs of 54 megabases or larger. That's unbelievable kinds of data to be able to look at this. And this is going to help us really resolve all those STRs and structural variants and any other kind of variation that we've been missing. Here I'm actually showing you a dot plot of the assembly here versus the reference, and it's astonishing to me that just pretty much out of the box, we're assembling full chromosomes like chromosome 11. And so I think as we begin to build up the biology side, we also need to kind of resolve the remainder, those last few remainder of patients that we just really can't find anything. And I think this is an excellent path for moving forward. So I will stop there, and it looks like we have some time for questions still. Thank you.
Thank you, Tychele. Wonderful. Questions for Tychele? So let me ask, so I saw that the enhancer was preferentially active, I think you said, in prenatal tissues.
Yes.
So in your model, have you thought about including the sensitive developmental periods in the expression of particular phenotypes?
We have not thought about that, actually. That's a really interesting point. If you don't mind, I'll just write that down.
Because there seems to be some, some data suggesting at least that this may play a role depending on when in a developmental period, a gene or a controlling element is active, can shape the outcome of a phenotype.
Yeah.
In the extreme cases, autism versus schizophrenia or intellectual disability, but there could also be more subtle changes. Or we have a
Will Keystone has hands up. I'm going to allow to talk. Will.
Hi, how are you? Thank you very much. Very interested in the subject. Do you have, is there an estimate of how many genes may be involved in autism? Some type of rough estimate, or in a person's cognitive abilities?
Yeah, so the estimates for that is somewhere between 500 and 1,000 genes, and that's been modeled by a few different groups. But I think that's a pretty reasonable number that people have come to. So we're at 300. We have another 200 to 700 depending on where we.
So that's okay. So basically, I identified. They feel confident about 300 genes, and the estimate's something 500 or 1,000 that may still be discovered. Is there any kind of estimate, like in a human's cognitive abilities, how many genes that might involve?
Yeah.
Anyone ever even thought about that?
Yeah, I think that the groups maybe more towards, so in intellectual disability, right, it's up more towards 1,000. But in terms of cognitive ability more generally, I would say that the people who've studied that the most would be the people, and again, it's a proxy, right? But like people studying educational attainment, like polygenic risk scores. I do not recall off the top of my head what the number is from that, but I would suggest you check out that work because there's been a few papers now on that. And I think there are some estimates there.
A follow-up, if anyone, what is the most accurate sequencing for exome sequencing that people are using now, or that you're using for your studies?
For exome sequencing?
Yes.
Well, I haven't done much exome sequencing in a while. So we mostly use for short read, this data I've talked about today from the New York Genome Center. And as we move to new technologies in the lab, we've looked at PacBio, ONT, BioNano, 10x, and they all have different good qualities. So I will hold off my judgment on that at the moment. They all each do something really good, and I have an inclination on what probably is the best, but I have a little more testing to do.
All right, I appreciate it.
Yeah.
Thank you very
Richard, you can go ahead.
Hi, Tychele. This is Richard Chen. Thanks for this really excellent talk. And I see you've been really busy in that beautiful building of yours. So I'm not a geneticist, so I'm going to ask you a question, but I think this is a general audience, and that's probably okay.
Yeah.
I love the slide you showed, where you sort of summarized work from your own lab and put CHD8 in a hierarchy that was under EBF3.
Yes.
The idea being that you can look up and down this hierarchy and see familiar players. So that gives you comfort. And not only that, but you can see that they share the feature that they're male specific. And then you said something really intriguing to me. And that is that if you look at a sodium channel gene, SCN2A, you were maybe surprised and maybe even a little disappointed that it wasn't yet part of this family. Now, that could change, but let's say right now it doesn't look like it's part of this family. One interpretation of that is that it's a big family, and I'm using this metaphorically,
right?
And that SCN2A drives excitability. Excitability is related to synaptic function. There are a lot of synaptic genes involved, but those synaptic genes drive a lot of different branches of the family, including the EBF3 and the CHD8 subfamily, but let's say another completely different subfamily, and you just don't see them being related because the statistical correlation is washed out by the fact that it's related to a lot of other branches. Is that all possible?
I think it is. I think that our grasp of the networks in autism are good. I mean, there's been a lot of efforts in there, but they're just nowhere where they need to be. Like, I think there are two areas that are really important in autism. One is fulfilling the allelic series and all of the genes that we need to know, right, that are involved. But the other branch of it is figuring out where they are and what they're doing and at what time point they're doing it. And I think we have a long ways to go. So I think that you're absolutely right. It's very possible that SCN2A is somewhere there, or it's in a different part of the network we just don't see yet. And I would just note that I made that very simplistic model based on the data that we have currently just for this particular subset. So,
okay, but just to be provocative,
Yeah.
let's say it's just a working hypothesis.
Yeah.
So then you could look at SCN2A and ask, is it involved in HADS, HADDS? Is it male dominant, et cetera? And you might expect that there would be other indications that would, let's say, synergize with your indication that this is in a different part of the universe.
yeah, it's very possible.
But you don't know the answer to whether, for example, SCN2A is male dominant or involved in HADS or anything like that. We would need to go look that up.
SCN2A, as far as I know, is not male dominant. It's in both males and females. In autism, that doesn't mean there isn't another phenotype that I haven't looked at where it's male dominated. And as far as I know, in HADS, EBF3, in the HADS syndrome itself is only EBF3. There may be HADS-like syndromes that, could be similar, but no, HADS is defined to EBF3 at the moment.
Okay, thanks.
Yeah.
Ben Tycho has a question. Please go ahead.
Yes, I have an easy one. That was great. And which paper should the uninitiated look at this point?
To Autism Genetics.
For your work.
Oh, for my work. So we actually have a bioarchive preprint. I think I put it on this slide just for that main non-coding story. Let me just put it in. Can you guys see the chat box?
I can take a screen cap.
Okay, let me just try to find it here. Oh, it's right here. Actually, I have it. So it's right here at the bottom. Is this Patty at all? You can also just look up Evan Patty. He's the first author.
You're very modest with your publications there. I see the size of the font. Okay, thank you.
Sorry. Yeah. So yeah, you can look up my name or Evan.
Yeah. Okay.
Yep.
Tychele, can you, we have another question. I'll
Okay.
hold mine then.
I think Will had another question.
He's muted on.
I'm unmuted now. Can you hear me?
Yes.
Oh, thank you very much. You mentioned approximately maybe 300 identified genes possibly linked to autism. Are these genes presumed to be acting simultaneously, or 50 are there many different ways for the autism to be in combination?
Yeah, so these 300 are based on protein coding de novo mutations. Those would be from, I can actually bring up that screenshot of the protein that we studied here.
Thank you.
There are these two papers, this Coe and Kaplanis paper. I think Kaplanis is now in Nature as of a couple days ago. But these are genes that are identified for having protein coding de novo genome-wide statistical significance. And so those are genes that are by and large are identified as like the one gene in that individual. There are more complex, which I kind of alluded to in the genome section, where we have these oligogenic, where we have multiple gene mutations that are important. And then of course that builds all the way out to polygenic, which has, a much bigger signal, but then there's lots of genes and they're not so well defined. So these particular gene lists are specific to like rare variants of large effect is how you can think of them.
And meaning, are you saying that a person who has autism would have one of those genes for sure? Or are you saying they would have all of them?
No, they would have one. So for example,
at least one. All right.
CHD8 is one. But so when you actually go back and look at the patients, then you explain maybe 30% from these. So there are actually still a lot more patients. We don't know the gene. We don't know what the mutation is
Sure.
that is important. But for example, CHD8 was a really good example where if you see a loss of function in CHD8 that's de novo in a kid with autism, you would probably say that's probably what's important in that individual.
Okay, so possibly many different ways
Yes.
to get the autism
Yeah, it's very high
in the genotype.
Yep, high heterogeneity. That's been the real tough part of autism and other neurodevelopmental disorders is that heterogeneity. If it was just one or two genes, we would have found them very early.
Right. Yes. Yeah. Exactly. With a neurological trait. Yep.
Tychele, between the Novo mutations in coding and non-coding space and the PRS that is known today, how much do you think genetics of autism is explained with the current findings?
Yeah, I think that's a good question. The estimates for non-coding are kind of rough. So let's just say the common variation can, let's say, contribute up to 50%, let's say. And then these rare variants could contribute to another 30%. They're not actually all measured quite the same. So I'm kind of not speaking correctly here, but let's say it's at 50%. Let's just be conservative. Let's say we added the polygenic and the rare variants, and we know there's some overlap. So let's say it's at 50%. I think the non-coding adds another 5% based on the limited ways that we've looked at non-coding so far. If you wanted to be really generous, you could say we're up to 80%, but I don't actually believe that that's where we're at. So yeah, I would say the non-coding is probably another 5%. I would say other people have argued it's more like 1%. So it's a number that's up for debate at the moment. But I think it's more like 5%.
With that,
yeah.
Thank you. I think we're out of time.
Okay.
Thanks again, Tychele. Wonderful
Thank you.
story. Thank you.
Thanks for the invitation.
I wish Zoom had like a virtual applause or something. So thank you.
Oh, thank you very much.
Tychele.
Okay.
Thanks. Bye-bye, everyone. Bye-bye.
Bye-bye.